Hello everybody. Today we are here to talk about the magical enzyme sucrase. Human sucrase, also known as sucrase isomaltase, is a digestive enzyme that catalyzes the hydrolysis of sucrose into its subunits fructose and glucose, which you then use for energy after eating. What about the function of sucrase? Sucrase actually has an incredibly important role during the final stage of carbohydrate digestion. This enzymatic process occurs on the brush border of the small intestine during digestion. Once sucrose is broken down into glucose and fructose, these sugars can be absorbed by intestinal cells and then transported into bloodstream for energy use by the body. Let's talk about sucrase down at the gene level. The protein coding gene responsible for giving the instructions for making the sucrase isomaltase enzyme is the SI gene. Sucrase is part of glycosyl hydrolase family with a whopping length of 1,827 bases and a mass of 209,453 daltons. What about some additional structures that make sucrase unique to its function? Sucrase actually has 4 binding sites that can allow for substrate binding of sucrase and even 5 active sites where catalytic activity of sucrase interacting with its substrate occurs. Sucrase also has 5 disulfide bonds between position 63 and 646, contributing to the overall stability and strength of sucrase. All right, let's talk about post-translational modifications. Sucrase's precursor protein actually gets proteolytically cleaved when it is exposed to pancreatic proteases in the intestinal lumen. This removes the initiator methionine at position 1, which allows to activate the sucrase enzyme. Sucrase displays tyrosine sulfation at 7 different residues. Tyrosine sulfation essentially involves the addition of a sulfate group to the hydroxyl group of a tyrosine residue in sucrase, which typically occurs in membrane proteins involved in cell addition, receptor signaling, or protein-protein interactions. Sucrase also displays phosphorylation at just 1 residue at position 7, where phosphate group was added to sucrase, particularly by protein kinases on the side chain of specific amino acid residues like serine, threonine, or tyrosine. This process is very important for regulation of sucrase activity. Sucrase also displays glycosylation at a wide variety of residues where glycan chains are added to proteins. This process is actually quite important for the structure and function of these modified molecules. So what about the domains? Well, sucrase has 2 of them, P-type 1 and 2 domains. The P-type domains contain 6 cysteines linked by 3 disulfide bonds with connectivity 1 to 5, 2 to 4, 3 to 6, which leads to characteristics 3-leaved structure, the trefoil with 3 loop-like regions. The central core of the domain consists of a short double-stranded antiparallel beta sheet, which is capped by an irregular loop and forms a central hairpin at loop 3. The beta sheet is followed by a short alpha helix with majority of the remainder of the domain contained in 2 short loops which lie on either side of the central hairpin. Now let's talk about the exact mechanism of sucrase, which uses a hydrolysis mechanism for catalyzing products. The substrate sucrose, which is composed of bound glucose and fructose, comes in and binds to the 2 active sites on the sucrase enzyme. This interaction forms an enzyme-substrate complex. The binding of the substrate in enzyme puts stress on the glucose-fructose bond that make up sucrose and makes it eventually break. So this breaking of bond causes both glucose and fructose to be released from the enzyme. and this allows for the process to start all over again. In other words, sucrase catalysis, the hydrolysis of sucrose to produce glucose and fructose, then the brush border transporters facilitate the absorption of these two monosaccharides. Now to our primary article. This article titled Blood Group Activity of Human Sucrase from Intestinal Metaplasia focuses on the role of sucrase in a physiological context outside of just digestion, helping us understand potential connections between sucrase activity, blood group interactions, and the development of intestinal metaplasia, potentially leading to stomach cancer, and whether sucrase may have something to do with it. So what was the purpose of this study? Intestinal metaplasia is a condition involving the precancerous change of the mucosa of the stomach with intestinal epithelium and is linked to stomach cancer. Sucrase, an enzyme involved in digestion, is actually found to have variable ABO blood group activity, and it is sugar moiety, similar to cancer tissues. Blood group activity refers to the ability of sucrase to exhibit specific interactions with blood group substances, which are molecules present in our tissues that carry ABO blood group antigens. These ABO blood group antigens have specific sugar chains that determine a person's blood type. The study specifically compared sucrase in areas of intestinal metaplasia with that in the normal small intestine, with a particular interest in its ABO blood group activity and sucrase from areas of intestinal metaplasia. Now, why would sucrase have anything to do with cancer? Here is some evidence that may suggest a link. Intestinal metaplasia in humans resulting from chronic gastritis involves abnormal differentiation of the stomach mucosa, leading to the emergence of intestinal type epithelial cells. Enzymological studies have showed the presence of small intestine specific enzymes in areas of intestinal metaplasia, particularly sucrase. Since stomach cancer often originates from these areas, research has focused on understanding its role in stomach carcinogenesis.
So let's talk about methods. How did they do it? To begin, tissue samples from gastric cancer patients were obtained and analyzed for intestinal metaplasia. The ages, genders, and blood group types were recorded. Sections from the small intestine were obtained during gastric cancer operations from a patient, 61 female type B, and 2 colon cancer cases, 71 female type A and 56 female type O. The test tape method was employed to determine the area of intestinal metaplasia by isolating the mucous membrane from the submucosa, which was then stored at -70 degrees Celsius. Subsequently, sucrase purification was carried out, which involved solubilization with papain, ammonium sulfate fractionation, gel filtration on a biogel P300, chromatography on DEAE cellulose, and electrophoresis on polyacrylamide gel to separate any contaminated minor blood group substances. Sucrase activity was measured using Dogger's method, 0.1 molar sucrase as the substrate. Experiments on pH effects utilize a two-step method with a tris/glucose oxidase reagent and buffers of various pH values that all contain 0.1 molar sodium ions. For kinetic studies, 7 different substrate concentrations were used, and the Michaelis constants were determined by Lineweaver and Burke method. Protein levels were determined by the Lowry method. Polyacrylamide gel electrophoresis was conducted by utilizing a 5% acrylamide separating gel. Gels were stained for protein with Kumasi brilliant blue, or for sugar using the periodic acid shift method. Some gels were sectioned transversely, diluted phosphate buffer saline overnight, and analyzed for sucrase and blood group activity. ABO blood group activity was measured using hemagglutination inhibition test. The wells contained 25 microliters of antigen, diluted with phosphate buffered saline. After 2 hours incubation at 37 degrees Celsius, 25 microliters of glutaraldehyde fixed red blood cells were added, and the inhibition and titer endpoint was determined as the dilution for inhibiting half the hemagglutination. Now, let's move on to the results, starting with electrophoresis. The polyacrylamide gel was used for electrophoresis, and the results are quite interesting. Blood group activity was detected in 2 preparations of sucrase from the small intestine. The sucrase from a patient of blood type group A displayed hemagglutination inhibition of type A only, and sucrase from a patient of blood type group B displayed inhibition of B only. The sucrase from a patient of type O, as expected, showed neither type A nor B activity. Now, as for the blood group activities of the 5 enzyme preparations from the area of intestinal metaplasia, preparations from 2 patients of type A blood showed no type A or B activity. One of the 3 preparations of type B showed neither type B nor A activity, whereas the other 2 preparations showed type B activity but no type A activity. Here's the distributions of the 2 activities in the gels. The top one is a patient from the small intestine group, type B. The other 4 are patients from the intestinal metaplasia group. You'll notice that the blood group activity was consistently found at the top of the gel. Minor protein and sugar bands at that position were also detected, possibly indicating the presence of glycoproteins or glycolipids interacting with sucrase. Interesting, right? Again, we see that only 2 of the intestinal metaplasia group had similar blood group activity as the small intestine group. So, let's take a look at the measurements from kinetics and pH activity measurement. With sucrose as the substrate, the Michaelis constants from the purified enzymes from the small intestine and an area of intestinal metaplasia were similar. Their Vmax values were also similar, being 25.6 units per milligram of protein and 23.2 units per milligram of protein, respectively. The pH activity profiles of the 2 enzymes were also essentially the same, both peaking at a pH of 6. Despite the absence of blood group activity, as we mentioned earlier, those 3 intestinal metaplasia preparations exhibited similar enzymatic properties to sucrase from the small intestine. So, in summary of all that information we just told you, this is what it means. All patients of blood type A or B were found to secrete material with blood group activity in their saliva. 2 of the 5 enzyme preparations from areas of intestinal metaplasia had similar levels of blood group activity to those of preparations from the small intestine, and the other 3 preparations showed no blood group activity. Despite that fact, those 3 preparations still exhibited similar enzymatic properties as the small intestine sucrase. Now, a possible explanation for the lack of blood group activity in these sucrase preparations could be attributed to incomplete sugar chains. Normally, ABO blood group antigenicity is determined by a terminal portion of a sugar chain, and it is normally present in several organs besides erythrocytes. Recently, reports have shown that blood group activity is actually deleted in the stomach and colon tumors, and that the new antigens that lack blood group sugar chains will appear in these tumors. Some changes of membrane glycoproteins and glycolipids have been associated with tumor formation, which could have also been due to the incomplete sugar chains. Taking all that information into consideration, it could be a possible explanation for the lack of blood group activity. The study goes on to discuss that tumor-specific antigens have been found in gastric carcinoma, as well as areas of intestinal metaplasia. From this, researchers suggest that there may be a link between intestinal metaplasia and stomach cancer, and thus, sucrase may have a role in it. This study was able to explore the role of sucrase in a physiological context outside of just digestion, helping us understand potential connections between sucrase activity, blood group interactions, and the development of intestinal metaplasia, potentially leading to stomach cancer. Thank you
Thank you
so
all so
much
much
for
for
watching,
watching,
and don't forget to check out our references.